Okay, good afternoon. So the work in our laboratory addresses this long-standing question of how do we explain this enduring link between early experiences and adult well-being and emotional behavior. And we focus on the development of the amygdala as being part of the link between these two endpoints, because the amygdala is responsible for paying attention to emotionally arousing events and learning about what's safe and dangerous in the environment. So it's really well positioned to explain some of the adult emotional behaviors that we're interested in. But moreover, the amygdala is rich with stress hormone receptors, and it retains its developmental plasticity for a very long time, which really makes it a good candidate for understanding how the environment influences brain development. In adulthood, the amygdala has strong bidirectional connections with medial prefrontal cortex. And medial prefrontal cortex is a region that incorporates information from not only the amygdala, but various regions of the brain that can synthesize that information for the service of coordinating and ultimately regulating some of the overarousal of the amygdala. And we study this system during development because it allows us to ask how the child's brain is responding to changes in the environment. Now, the World Health Organization published a study from 21 different countries around the world showing that childhood adversities actually contribute to about 30% of all adult mental health disorders, and you can see the specific breakdowns here. Importantly, the authors conclude that these estimates might actually be conservative. So what that means is childhood adversity is actually one of the leading causes of adult mental health problems. So today, I'm going to be focusing on emotional neglect by the caregiver. And the reason to do this is emotional neglect is a very common form of childhood maltreatment. But unlike things like physical abuse and neglect, emotional neglect is rather invisible. It's harder to see some of the outcomes associated with this type of adversity, and therefore it's often overlooked by clinicians or teachers or, for that matter, the research community. It's often thought of as an absence of input, but this absence for a developing brain that's expecting a certain parental input is a major stressor for the developing brain, and in many ways, emotional neglect is a more pernicious form of childhood maltreatment. There's a failure to receive the needed parent-child intimacy, support, and serve and return that are critical for brain development. And so often children are described as raising themselves to a large extent, which is really a significant stressor during brain development. So if we consider the absence of parental input as a major stressor, we can turn to the basic neuroscientific literature that's shown us that at the single cell level, the amygdala is very responsive to chronic stress. So the amygdala cell tends to exhibit amygdala hypertrophy and greater reactivity, which really, benefits the individual for identifying and dealing with future stressors or responding to future stressors in a faster manner. But that's in stark contrast to what's observed in the neighboring hippocampus, which Kim just talked about, where chronic stress tends to result in decreased dendritic complexity and smaller cells. So this contrast between the amygdala and the hippocampus starts to give us a clue about how stress is impacting the brain. It's not impacting the brain in a uniform fashion, but rather it's informing the brain about how to respond faster to stressors in the future, but perhaps at the cost of other processes. So when this stress happens during development, one of the reasons why it's so potent is that those stressors are co-occurring with a sensitive period for development of the amygdala and its connections with prefrontal cortex. So I just want to briefly share with you some of our findings in typical development that supports that view. So when we look across age here, we find that the amygdala has a very robust response to emotional stimuli like fear faces at younger ages, which makes a lot of sense because you're learning a lot about the emotional world at this young age. So despite this early reactivity that we see in the amygdala, we see a very delayed development of the connections between amygdala and prefrontal cortex. So what I'm showing you here is in the adult, we get this nice anti-correlated pattern between amygdala and prefrontal cortex. So when the prefrontal cortex increases its activity, the amygdala decreases and vice versa. It's very consistent with a regulated pattern, emotion regulation. In older and younger adolescents, we still see that anti-correlated pattern, but you can appreciate that the magnitude is greatly decreased, consistent with what we know about the slow development of emotion regulation. But what we were most interested in is in childhood, we didn't see evidence of that adult-like regulated pattern. We saw this positive connectivity. So that's to say that when children are on their own, they're not exhibiting this intrinsic means of emotion regulation at the neurobiological level. So taking these data together, if this is age on the x-axis, what we find is that the amygdala exhibits this strong reactivity early in life, but that's happening in the absence of these adult-like connections with prefrontal cortex, which we don't see emerging until the beginning of adolescence, which really makes childhood a very good candidate for looking for these sensitive periods in the brain when the environment can influence future development. Moreover, we have evidence to show that the nature of the relationship between amygdala and medial prefrontal cortex during childhood is a very good predictor of how the system stabilizes over time. So Sonia Lupien and colleagues have noted that it's the period of the most rapid change of brain development that heightens the opportunity as well as the vulnerability of that particular brain region to environmental exposures. So we've since become very interested in the environment in which this system is developing. And so it's certainly not developing independently in a test tube, as our MRI scans often lead us to believe, but it's developing in a very special species-expected environment, which is the parent. And in the human, the parent can be the mother, the father, the grandparent. This is a learned relationship. And humans certainly invest a lot of time into caring for their young. And this has led to notions like there is no such thing as a child, which is just a reminder that in order to understand human brain development, we really have to understand the environment in which that brain is developing. So I like to think of parents as providing a scaffolding for brain development, much in the same way that buildings during construction have scaffolding that direct its future growth. So we turn again to basic neuroscience to address this question of the intimacy between stress-reactive neurobiology and the parent. So this is work by Regina Sullivan, and this is done in rodents. But briefly, what her group has found is that there's this period during development where amygdala functioning is critically dependent on the parent. So when the parent is available in the nest, the amygdala actually remains fairly quiet, and that happens by the parent reducing stress hormones that would normally activate the amygdala. But when the mother is out of the nest, the amygdala engages and contributes to fear learning. So how that plays out is, in terms of behavior, is striking. So the workhorse of threat learning or aversive learning in neuroscience is aversive conditioning. So for example, in this group, if they pair a peppermint odor with a mild foot shock, and this animal learns this on its own, and you place the peppermint odor at one arm of a Y maze, the animal does more or less what you and I would do, and it will avoid that peppermint odor. And that's what's shown here, very few choices towards that peppermint odor. But if the learning happens in the presence of the parent, and the parent can quiet the amygdala, then you don't get avoidance learning. The parent blocks avoidance learning and instead promotes preference learning. So the animal will approach the peppermint odor. And if that sounds strange to you, this is actually developmentally appropriate because the animal at this young age doesn't have the ability to fight or flee from a threat. That's not a survival strategy at their disposal. Instead, attachment is the strategy. And so the idea is that early in life, the presence of the parent helps the preference learning systems or the attachment promoting systems trump avoidance learning or trump fear learning. So we were very interested to know whether similar learning mechanisms occur in the human. And so we presented young children with a conditioning paradigm where the blue square would always co-terminate with not a shock, but an aversive noise, which is like nails scraping down a chalkboard. And the purple triangle was paired with nothing. Children learn these associations either sitting next to an experimenter or sitting next to their parent. Then the parent leaves, and we place children in a human Y maze. So in the human Y maze, children enter a tent, and they see the blue square here and the purple triangle here, and they're asked to pick a door to retrieve a prize. And they're shown that the same bucket of prizes is behind both doors, but just pick a door, and they do that 5 times. So what we find in about 100 3-, 4-, and 5-year-olds is that when children learn these associations by themselves, they act more or less like you and I would. They're avoiding that blue square. But if the conditioning happened in the presence of their parent, then the avoidance learning is blocked, and although it's slight, there's a significance preference learning in the human as well. So importantly, these are the same children whose behavior is switching as a function of the learning conditions. So this suggests that similar learning is happening in the human. So we had the opportunity to collect MRI images while children were presented with cues of their parents, so pictures of their parents' faces, and contrasted it with somebody else's parent's face. And what we observed is during childhood now, as opposed to that heightened amygdala reactivity, children are actually showing a decreased amygdala response. So this is consistent with the notion that parents are effective buffers of this hyper-amygdala response that we typically see in childhood. Parents' stimuli are also effective in strengthening momentarily that anti-correlated, that adult-like pattern between amygdala and prefrontal cortex. So in other words, without the parents, children are exhibiting this childlike, non-regulatory pattern of connectivity. But with their parents, physically, they instantiate this more adult-like regulated pattern between amygdala and medial prefrontal cortex, and the strength of that is moderated by the child's attachment security with their parent. So what we are thinking is happening is that parents are effective in modulating this stress reactivity. Parents go away, they go to work, children go to school, and then they come back together. And that routine going away and coming back together is really important in toning or in training the system in how to operate once independent, so once the scaffolding comes down. So we've had the opportunity to ask the question about, so what are the effects of the absence of a parent, so the parental neglect? And we've scientifically pursued an extreme form of this type of neglect, and this would be early institutional care, so what we commonly call orphanages. And in the samples that we've worked with, all of the children have since been adopted by families, and this is adoption via international adoption. And I point that out because the families that adopt internationally, so in Environment B, are a very special group of families who we infer have a great desire to provide care for their children because it's not easy to adopt internationally. So the environments A and B are drastically different from each other in terms of parental input. So even in the best case scenario, even with the best of intentions, institutional care is a far cry from family environments. So children show significant initial developmental risks. However, they also show significant rebound in several domains of behavior upon adoption. As I'm going to show you, there's tremendous heterogeneity. So I think that's really important to say that children who experience different forms of adversity are at significant risk for poor outcomes, but there's development is not deterministic, right? So there's a lot of individual differences that I'll be pointing out. But the most common risk that we hear from parents is difficulty with emotion regulation behaviors. So the data that I'll show you include participants who were first placed in institutional care before their 6-month birthday and adopted by their 2nd birthday. So when we look at things like internalizing problems and externalizing problems, so internalizing problems are things like anxiety and depression. Externalizing problems are things we often think of like attention or difficulty with inhibitory control. We see that previous institutional care is associated with a significant increased risk for both of those with large effect sizes. But I also want to point out that there's a lot of individual differences. And so much of our work is trying to understand what are the factors that differentiate the kids at different levels of risk, and how can we understand how to reduce the risk for the children who are at the highest levels. So using structural MRI, we've observed that a history of institutional care is associated with a larger amygdala volume, but only for those children who had spent a year, at least a year in institutional care. Now, importantly, this correlated with internalizing problems and trait anxiety. So it was the children who had the larger amygdala volumes that were showing the most difficulty in these behaviors, whereas the children who didn't show those amygdala increases were not showing those same behavioral outcomes. In response to things like fear faces across 2 separate cohorts, we've identified elevated amygdala excitability following this type of early experience. But again, turning to the basic neuroscience, we were really struck with a number of papers that have shown in rodent models, for example, that stress during development seems to do something different than stress in adulthood. During development, stress actually seems to accelerate development of some of these stress-reactive neurobiological systems. And that was surprising to us at first, because very often in the literature, there's an association between stress and impaired function. And here, the suggestion was that stress during development may actually be facilitating some processes. And the idea is that when differences in behavior and brain occur following early stress, they may be best understood as developmental adaptations. Those are adaptations that happen during development in order to meet immediate needs. But the question is whether there are potential trade-offs to consider. So are there longer-term costs that we want to be looking out for? And so we have some evidence from our lab that something similar may be happening in children who experience parental deprivation. So these are the amygdala prefrontal connectivity data I showed you at the beginning of the talk, where children with a typical caregiving history show a more childlike, non-regulatory pattern that switches to a more adult-like regulated pattern in adolescence. We were able to replicate that finding in a separate cohort. But when we look at this connectivity pattern following early institutional care, in childhood now we don't see that childlike pattern. We now see a more adult-like pattern of connectivity between amygdala and medial prefrontal cortex. So in other words, a 6-year-old in this group has a pattern that might look more like a 16-year-old. So this effect is mediated by the stress hormone cortisol that we collected in the laboratory, but we're also understanding from the behavioral outcomes that this too may be an adaptation in children. So what we think is happening is that in the context of amygdala hyperactivity that results from this early potent stressor, the connections between amygdala and medial prefrontal cortex may accelerate through an activity-dependent process. While that may confer some benefits, the potential risk is that we might have abbreviated the sensitive period of childhood. So here are the data that lead us to think that this might be an adaptation. So although at the group level, early institutional care is associated with greater anxiety, the children that exhibit this more adult-like connectivity pattern have decreased anxiety relative to peers who did not exhibit that pattern. So to say it another way, the children here have a brain pattern that's most different from the typical group, but that difference seems to confer some benefits in terms of emotion regulation abilities. But on the flip side, we also want to ask, if acceleration abbreviates plasticity, would that place limits on the ability of the amygdala to be buffered by parental stimuli? And at the group level, that's what we find. So these are the data I showed you earlier with the parent-stranger paradigm, where children with a typical caregiving history show parental buffering. At the group level, we don't see that same pattern following early institutional care. But I want to point out that these error bars are very big, which means that some of these children are showing evidence of parental buffering and some aren't. So we wanted to interrogate those individual differences over the long term. So we were able to follow children for over 2 years in this study. And what we found was that within the group that had experienced early institutional care, exhibiting that parental buffering at time 1 actually helped bring down anxiety over a 2-year period. And when we asked, well, what was different between those kids that showed buffering and those who didn't, it was the factor that differentiated those was the child's reported attachment security with their parent. So in other words, the children who exhibited amygdala buffering at time 1 also reported having more feelings of security with their parent. And I think that's really important because it suggests that if we're going to care about children's brain development, we also have to care about the family and support the family in order to enact those changes. So to conclude, there seems to be a long sensitive period for environmental influences on amygdala medial PFC in childhood. So this gives us the opportunity to think about how the environment is scaffolding development. Parents are really primary social regulators of this amygdala PFC development. So parents really provide a privileged information source over and above other inputs. Early caregiving adversity does significantly increase the risk for poor mental health, but there are large individual differences, and I think those are meaningful and important to understand how to enact change. And importantly, strong parent-child relationships have powerful healing effects well into late childhood and adolescence, as our data would suggest. So lastly, we think that early caregiving adversity may have its influences on the nature of behavioral and brain development by perhaps influencing the pacing of development. And so understanding brain development through the lens of adaptation may be the best route to understanding pathways towards change. So with that, I'll thank the funding, the members of the lab that contributed, and the families and individuals that participated, and thank you for your attention.